This section explains why the cell is not where life begins and what sits beneath it. The cell is usually taken as life's atom. Weiss put it plainly, a building consists of bricks. Chapter 1 removes that floor. Bacteria and blue-greens, which Haeckel called cytodes, live and reproduce, yet lack the cell's defining machinery. No nucleus, no chloroplast, no centrosome, no organelles. Their body is a bioblast, a simpler order of living thing that sits below the cell. The move is subtractive, and it is the step that does the work. Demote the cell from the minimum of life so it can later be rebuilt as a composite. Fix the elementary unit below the cell, and the eukaryotic cell stops being primitive. It becomes something assembled, a consortium. That single deletion is the whole book in miniature. This section explains the elementary unit that sits below the cell. The cell is not the floor of life. Below it sits the bioblast, the bacterium, the blue-green, a living body that lacks nucleus, chloroplast, centrosome, every organelle the word cell exists. Kozo. Polyansky's opening move is a refusal of analogy. The bacterial body is a refusal of analogy. The bacterial body is not a reduced cell. It is a lower stratum of organization entirely, and the controversy over what its granules are only proves that no honest correspondence to a real cell exists. Naming this pre-cellular unit is the chapter's whole function in the arc. It fixes the elementary part that every later chapter will assemble by symbiosis into the composite eukaryotic cell. The floor beneath the floor. This section explains why some parts of the cell are best understood as guests rather than manufactured parts. What looks like an organ of the cell is often a guest, an organism that moved in and stayed rather than something the cytoplasm produced. The whole work of the chapter is to break one word, organelle, apart. One meaning is a part that arose when the cytoplasm differentiated internally. The other is a form of free-living organism that moved in and stayed. Kozo, Polyansky picks the case where the seam still shows, the green and yellow chlorophyll organelles of animals and protists. These turn out to be whole algae, the zoochlorellae and zooxanthellae, each with its own wall, nucleus, and chromatophore, and each culturable outside the host. The diagnostic is autonomy. It divides on its own. It can be expelled and still survive. Establish the inference on the case that separates cleanly, then aim it at the plastids that no longer do. This part explains why a green granule inside a cell is read as a captured organism rather than a piece. The cell grew. The green granules that let an animal photosynthesize were read for a century as parts the cytoplasm grew until their autonomy gave them away. Each keeps its own wall, its own nucleus, divides on its own schedule, outlives its host, gets expelled when its own schedule outlives its host, gets expelled when starved, and can walk back into free-living life. A true part cannot leave. A guest can. So the organelle is captured rather than manufactured, a whole organism engulfed and kept, then inherited equally by both daughter cells at division. This is Kozo. Polyansky's generative move, read a component's independence as the fossil of a former symbiont. The chloroplast is an algal cell that never went home. This section explains what it means to call something an individual and why the boundary is drawn by the observer. An individual is a boundary drawn around a consortium. The lichen makes the move visible. Fungus and alga, each culturable on its own, fuse into a single body. That body has a thallus, a chemistry of its own, including licanin and roughly 200 lichenic acids, and a hardiness that none of the partners owns alone. Grafts and their chimeras push the point further. Scion and stock wrap around each other, close as a glove on a hand. Yet each parent can still be released. The autonomy stays latent rather than erased. The chapter's thesis follows from this. Multicellularity is symbiosis scaled up. The many-celled organism is a stabilized colony of once independent lives, and the one is a compression the observer imposes on a persisting several. This section explains why the multicellular body is best read as a consortium rather than an individual. The body is a consortium, not a single individual. The same symbiogenic move the cell chapter made, lifted one level up. The lichen carries the proof. Its two partners, an alga and a fungus, can each be cultured fully apart. Together, they produce a thallus whose architecture, hardiness, and reproduction belong to neither partner alone. Composition, rather than modification of a single lineage, generates the new whole. Its emergent properties live at the seam between two organisms, never inside either one. Predict the next case from this. Wherever a tissue resists reduction to one ancestry, look for a second lineage cemented into it and read the body as a truce rather than a thing. This section explains the philosophy of symbiogenesis, the argument that the whole organism is read backwards from its parts. 19th century biology builds the organism top-down. One homogeneous body differentiates into organs under a drive toward division of labor, a machine, and a machine needs a maker. Kozo. Polyansky reverses the arrow. The organelle came first, autonomous, a life on its own account. The whole is only what those partners collapse into after incorporation. Differentiation does not produce the parts. The parts, joining, produce the division of labor. A cell becomes a system, and a multicellular body becomes a system, and a multicellular body becomes a federation of organisms, not a colony. The chapter's real payload is the dissolution that follows. If new forms everywhere arise by combination in leaps, sulfur and iron making a crystal, then the organic and inorganic boundary vanishes and no maker is needed. This section explains the single inversion at the root of the whole chapter. Read the body as assembled, never as differentiated. That one flip generates everything that follows. Cell theory says a homogeneous body split into organs driven toward a division of labor. Symbiogenesis says the opposite. Formerly autonomous beings came together, and only their joining made any division of labor possible. The organism is a federation of self-sufficient partners that predate it, and that still reproduce, struggle, and vary, each on its own, rather than a machine built from the top down. Kozo. Polyansky's motto compresses the flip in the phrase. From section 10, partocentrism instead of totocentrism. Attention moves from the whole to its parts. Every corollary is this same move applied again, heredity, selection, and the tree of life that now, anastomosis, its branches rejoining rather than only splitting. This section explains how a theory of symbiogenesis earned its place by choosing which thinkers count as its ancestors. Kozo. Polyansky sorts every precursor by epistemic mode, by how each one reached the idea, never by whether the conclusion happened to match his own. Empedocles guessed the shape, imagining parts that combine before wholes, but he reached it from pure fantasy, since no supporting facts could yet exist. Oken is disowned as a category error. His so-called infusoria were merely the cells of the nascent cell theory. Darwin alone counts as the true progenitor, because he reached the same idea inductively, dragging indirect proof behind it. Then comes empirical closure, the zoochlorellae, the artificial decomposition of lichens, an independent organism shown to be an organelle. The chapter works as a legitimation genealogy. Its central axis is Aristotle's whole, before parts set against parts compose the whole, with symbiogenesis crossing from speculation through induction to experiment. This section explains why the fifth body of the chapter reads as a validation curve rather than a chronicle. The whole chapter is one claim, restated for 2 millennia, climbing only in how much evidence supports it. The invariant is this: a complex organism is an association of smaller, independent organisms. That claim recurs from Empedocles to the present day, differing only in how much proof backs it. Empedocles states it as pure fantasy because no facts were yet possible. Aristotle inverts it, putting the whole before the parts, and reigns unchallenged for centuries. Darwin, the real progenitor, reaches it inductively through heredity and grafting, yet never specifies the manner of combination. Entz and Brandt supply the direct proof, and Innes and Brandt supply the direct proof, an independent organelle. The names here are rungs, not events. What earns the idea? Its sciencehood is the monotone climb from speculation to demonstrable fact. And yet it moves, eppure si muove.